Steps in the replication of viruses. Viruses are not cells, but they require cells. As obligate intracellular parasites, they must attach to and enter a host cell in order to reproduce. Although different groups of viruses have evolved diverse strategies to replicate themselves, it is possible to review the events in the life cycle of a generalized virus to provide a background for understanding individual virus groups. Virtually all viruses must accomplish 8 key steps in their replicative cycle. Step 1. The virus attaches itself to its host cell. Step 2. The virus or its genetic information penetrates the cell. Step 3. The nucleic acid is uncoated, which frees the DNA or RNA from its capsomeres or lipid envelope and permits the host cell to read out the genetic functions of the virus. Step 4. At this stage in the life cycle of many viruses, only a portion of the viral genetic information is expressed, resulting in the synthesis of only the subset of viral encoded proteins collectively called the early viral gene functions. These proteins may function in one of several ways. In some cases, they contribute directly to the replication of the viral chromosome. In other cases, these viral proteins turn off many of the host cell activities, maximizing the cell's available resources for virus production. Alternatively, some viruses that can duplicate themselves only in actively dividing host cells produce proteins that stimulate host cell division. Step 5. The viral nucleic acid is then synthesized to produce hundreds or thousands of copies of the viral chromosome. Step 6. At this time, a second subset of the viral genetic information, commonly termed the late proteins, is expressed. These are the structural proteins including the capsomeres of the virus. Step 7. The capsomeres are assembled to form a shell around the nucleic acid of the virus. Step 8. The mature virus, having duplicated its new copies, is released from the infected cell to attack a new cell and repeat this process. Going from steps 1 through 8, a viral replicative cycle displays temporal organization. Specific events occur in sequence, each dependent upon the successful completion of the previous step. This developmental process, which results in the synthesis of thousands of viral particles, is formally similar to the development of the multicellular organism from a single fertilized egg cell. Both processes require quantitative and qualitative changes over time. Early viral proteins made from single genome of the infected cell are expressed at low levels, while the late proteins are made from the newly replicated viral chromosomes produced in step 5. Quantitative changes are thus initiated and regulated chronologically. And those are the steps in the replication of viruses. Have a great day.